it's been decades that we've had papers highlighting the inadequacies of some of these tests. If it doesn't work, it doesn't matter how cheap or convenient it is.
Professor Jenny Rohn is head of the Centre for Urological Biology in the Department of Renal Medicine in the Division of Medicine at University College London. Her lab focuses mainly on understanding the science behind urinary tract infection, examining bacterial behaviour and treatment failure using advanced human tissue models. She also collaborates with engineering, clinical and industrial colleagues to develop novel therapeutics. Thanks so much for joining me today to answer the questions submitted by our community.
Thanks for having me on.
I'm excited to have you on because your name comes up a lot in our emails and in our socials. So it'll be great to hear more about your research. But first, can you share more about your background and how you became interested in UTIs?
Sure. Well, I was, as you could probably tell from my accent, I was born in America. I was trained there. I got my PhD there. And I came over to the UK really interested in how our human cells react to various stresses and strains. And I spent quite a bit of time studying cancer. But I became a little bit disillusioned because there were about 10 gazillion people studying cancer around the world. And I noticed that there were other diseases that hardly got a look in. And when I found out about urinary tract infection, I was really interested in the biology of it because, as we may discuss later, it's quite complicated how the bugs attack the bladder. And when I started looking into it, I thought, well, great, who's studying this? And I did a Google search, as you do. And I discovered at the time, this is about 2010, I could only find about 12 labs in the entire world working on UTI, sort of at the molecular level. I thought, this is ridiculous. And I thought I saw my calling. I thought this is a really great opportunity for me to apply my interest in cells, human cells, with something more worthwhile and something that wasn't being done a million times by lots of other people. So that's how I got interested. And also as a woman, I'm really passionate about female health and particularly the way it seems to have been marginalized in research and also in clinical trials. So it sort of vibed with my feminist sort of outlook as well.
Well, that's what we like to hear. And it has become such a big problem that we really do need more research in this space. And it's always great to meet researchers who are focused on this. And today, we want to talk a lot about testing. So we got many questions about that. So let's just jump straight into it.
Great.
What are the main reasons that urine dipsticks and standard urine cultures miss so many infections?
Gosh, that's a sort of a very complicated question. There are a lot of factors. Let's just go through them. So first of all, when you get a urine dipstick or a urine culture, you're looking at urine. And if you think about your typical person who's drinking lots of water, perhaps especially if you have UTI symptoms, the first thing that they tell you if you call 111 is drink a lot of water. So by the time you get to the doctor and you produce your urine specimen, it tends to be highly dilute. And both of the tests that you mentioned, the midstream urine culture and the dipstick, are based on concentrations of stuff in the urine. So if you've just drunk 3 liters of water, you're not going to test very strongly on either of these tests. And the dipstick has particular problems. I think it measures 3 things that could be relevant for a UTI. It measures whether you've got the presence of white blood cells in your urine, whether you have the signature of particular bacteria like E. coli or Klebsiella. These are particular kinds of bacteria. It doesn't look at all bacteria, only a particular kind. And then also whether you might have trace elements of blood in your urine. So those are the 3 main things you would look at in a dipstick. All these are quite insensitive anyway. And then you've got your highly dilute sample, and you'd be lucky if you would twig on any of those 3 parameters. And then if you don't have the right kind of bug, for example, if you've got an enterococcus or staphylococcus or something like that, that's not going to even be picked up by that bacteria pad. So already you're looking at a situation where the deck is stacked against you. If you're lucky enough to get a positive dipstick and your doctor believes that you need to have a urine culture, you've got the same problem. Highly dilute urine will have fewer bugs than concentrated urine. And also the samples that go off to the lab, they might sit there for a while. They get sort of cultured under conditions that are very, very good for particular bugs, again, like our friend E. coli, maybe not so great for other kinds of bugs.
Hmm.
So you may not reach that magic threshold, whatever that might be. Usually it's 100,000 bacteria per mil. Some labs do less, but there's always a threshold cutoff. And if you haven't achieved that for whatever reason, in some cases because you've drunk too much water, then you're going to get a negative test. Your test might also get thrown out if you've got a mixed growth. So that's when you test for more than one bug. Historically, this has been seen as, oh, there's something wrong with the sample, let's just chuck it in the bin. Whereas we now know that it's highly biologically possible that you can have more than one bug causing an infection, or more than one bug can be hanging out in your bladder, and it doesn't invalidate the test. So these are some of the reasons why both of these tests aren't very good.
Right, and there's multiple problems. So it wouldn't be enough just to lower that threshold to cater to the concentration issue. We're just not picking up some of the organisms.
Yeah, the problem with that, of course, is that we now know that the healthy bladder contains a microbiome, not surprisingly, as the urethra is open to the environment and these bacteria they go anywhere. Bacteria will go anywhere where they can get. And you start getting into the territory of low-level, friendly bacteria that are in there. So once you have a very low count, there is the danger that you might start picking up the normal microbiota. And in that case, then that also complicates things because you might have you're more likely to have a mixed growth, which is going to be chucked out, or you actually don't know if it's a pathogen or not.
Right.
So I can see why you need a threshold, but it's a really difficult problem.
Yeah, it's something that a lot of people in our community have come up against, the negative test results despite their symptoms of UTI, even if they're very familiar with what a UTI feels like. And so it's constantly a source of frustration. But has there been much published on these tests and how inaccurate they are?
There have been papers. There's been quite a few papers throughout the history of the science. And I have to say that I find sometimes the medical profession is a bit conservative with a small c. So it takes a lot of research until findings from science filter down into medical practice. And that's probably a good thing because you don't want to rush into practice things that don't work, that aren't cost effective, especially with the national health where money is a serious problem. So yes, I can understand why it takes a while for these things to trickle down, but it's been decades that we've had papers highlighting the inadequacies of some of these tests. But since we've got nothing really better, I think and especially the dipstick. The dipstick is compellingly lovely. Like it's this little piece of paper. It's cheap as chips. It takes like 30 seconds. It gives you an answer, yes or no. And I can see why that's very compelling. But equally, if it doesn't work, it doesn't matter how cheap or convenient it is.
Yeah, and it leaves patients in this situation of not being able to get treatment because there was nothing to base the treatment on, and then also not being able to get better testing in order to find out what is the problem,
Yes.
even if it isn't an infection.
I understand that the dipstick is often a gateway to getting a midstream urine culture. And I guess in that case, it could prevent you from getting another test, although that test isn't that great either.
Yeah, exactly.
But it's probably a bit better than the dipstick, for sure.
Yeah. And with regard to the quality of a sample, do you think a catheterized sample is actually better than a midstream urine, or does that run the risk of then missing infections lower in the urinary tract?
Well, we don't know much about, if you have a UTI, it's really difficult to say where these bugs are. They could be in the bladder, they could be in the urethra. There haven't been a lot of studies about sort of looking at the different niches of UTI. It's always presumed that the bugs are in the bladder. But of course, they would maybe in the process of ascending, right? They have to come from somewhere. They're coming from the gut, ultimately. So they could be in there. If you have a catheter specimen, I mean, one of the problems is, of course, catheters can introduce the risk of getting a UTI. So if you get first of all, it's incredibly uncomfortable. If you've ever had one, you'll know it's excruciating. I don't like urinary catheters, but if you get one, so you could be damaging the tissue and promoting infection. You could be introducing new bugs by doing that. And also, I mean, we and others have done studies showing that, if you use a catheter, you can see bugs. So we know they are in the bladder, but I don't know if it's any better. Maybe it's nice to have the whole niche there, including the urethra.
Most patients would prefer not to have a catheterized sample if they can avoid it.
Right?
Yeah.
Yeah.
Well, I guess there's a lot of problems with testing. And so sometimes patients just end up getting treatment based on no testing because it's empirical. If the antibiotics do then work in that case, would you say that's a good indication that an infection was present?
I would have thought so. I mean, we can discuss the caveats here, which is, of course, the placebo effect. So, you're getting a drug, you might feel a bit better because of that. Just what they call sort of regression to the mean, or that's a
Hmm.
fancy way of saying, well, maybe you were going to get better anyway. You've taken these pills and you've gotten better. It doesn't prove you had an infection. But if you feel really a lot better quite quickly, it is a good indication that
Hmm.
there probably was something there, but you can never know. You never know.
That's true. I think a lot of patients can probably tell. And that's one of the
Of
things that we
course.
come up against, that the patient-reported symptom is not taken very seriously in research. And so I think that we really need to put more weight on that given the lack of accuracy in testing.
I mean, we do see in clinical trials that I've seen, things are getting better. So there's almost
Hmm.
always a quality of life assessment. So before and after treatment, you're asked to fill in a questionnaire about how you feel, what your symptoms are, what's your day-to-day life like. And so patient quality of life is being taken increasingly seriously as an endpoint in clinical trials, which is great, right?
So
Because
important.
nobody ever used to care. But
Yeah,
any
exactly.
self-respecting researcher is going to have that kind of patient-focused output. It maybe you're testing whether the bugs have gone down or you're testing whether the white blood cells have gone down. But if you don't test if the person actually feels better,
Yeah,
how good is that treatment?
not
So
very
yeah, I
helpful
think I
for
think
patients.
things are moving in the right direction. I think quality of life assessments are taking more and more of a center stage with outputs for
Yeah,
trials.
it's really good to see that change. When it comes to different technologies of testing, what are your thoughts on more advanced methods like next generation sequencing? Do you think they can provide useful information?
So the problem with next generation sequencing is, first of all, it's horrendously expensive.
Hmm.
And at the moment, we don't have one in every GP surgery, and it's used in hospitals. But we get the same problem that we have
Hmm.
with when we were discussing the microbiota and the sensitivity. So if you do next generation sequencing on anybody's urine, you're going to find bugs. There
Right.
will be bugs in there. Even if you're a man, there will be some bugs. So then you have this long list of bugs,
Hmm.
and then you're asking yourself, what's the pathogen? Is it necessarily the one that's got the most?
Right.
Or maybe there's one of those guys in the list is doing the job, but he's just more effective at it, so he can cause
Hmm.
more symptoms at a lower level. We don't know enough about the normal signature of the microbiota really to be able to just look at these printouts and say, oh yes, this is the signature of a perfectly healthy person, or oh no, this is the signature of a chronically infected person, and that is the culprit.
Yeah.
So until we do enough studies to understand like the vast variety of ecology that's seen in different people's bladders, it's going to be really hard to use that tool,
Hmm.
I think.
Yeah, we get that question a lot about whether something on a test, how do you know that it's actually causing the symptoms just because it's there? And in that case, a lot of people get high lactobacillus on certain test results. And does that mean the lactobacillus could be causing a problem? Or do we just not really know what's causing the problem, like you said, from a long list?
Well, if it's lactobacillus, it's probably friendly.
Hmm.
So lactobacillus is the most common bacteria in the female uromicrobiota. So you would expect to see lots of lacto up there.
Yeah.
And if I saw lacto in my bladder, I'd be thrilled because I would assume it's probably doing something good, or at least it's neutral.
Hmm.
Lacto plays an incredibly important role, it's thought, in protecting especially women from UTI, usually in the vagina, where it's sort of the last outpost of resistance before you hit the urethra.
Hmm.
But we don't really know what they're doing in the bladder. They may be passive passengers, or they may be actually protecting the bladder from UTI. That's one of the things that our lab's actually looking into now.
Oh, yeah.
Really interesting stuff. We don't have the answer, but I think, yeah, but your example is great. Like if you get massive amounts of lactobacillus, is that causing a UTI?
Hmm.
I kind of guess not, but again, who knows? Because bacteria never stop surprising me. You might think it's a friendly bug, but maybe it's gone rogue.
Yeah, for some people, it could be the problem.
Exactly. So I don't want to say, never, but I wouldn't be too worried about that. But then, Yeah.
Hopefully, for most people, that is the case, that it is protective, so we don't need to worry about that. And there's also a question often brought up about interstitial cystitis. And do you think that it might be the case that that is actually a missed infection given the problems with testing, or a biofilm or embedded infection that's just not being picked up?
I mean, the definition of this disease is usually includes excluding infection. So if you've got
Hmm.
no measurable infection, the algorithm would tell you to go down another route, which could include IC. I don't know much about IC, to be honest. It's not my
Hmm.
area of expertise, but of course, if you have symptoms and no measurable bacteria and the tests aren't very good, these people might have a hidden infection, of
Hmm.
course. But there are other options. They may have no infection and it's caused by something else. That is very possible because this is an inflammatory disorder. It causes lots of things can cause inflammation.
Hmm.
And then there's also some evidence that if you have a UTI and then you completely clear the UTI, you could still have symptoms. There's evidence from humans and there's evidence from mice. So some of these people that aren't responding to antibiotics, it's possible a subset of them are just hit and run. Post UTI, it's kind of like the
Hmm.
post-viral thing, like long COVID. You can get infections and then afterwards the infection can be long gone and you can still have symptoms. So that could be another thing. It's like the body was reacting to the bacteria, reacting
Hmm.
so strongly that even after the bacteria got cleared, it didn't get the memo to stop reacting. And I think
Right.
that could possibly explain some of the unfortunate people that
Hmm.
don't respond even to long-term antibiotics. But there's so much we don't know. And of course, mice are not people. We can do all
Yeah.
sorts of clever things with mice. The good thing with mice, this is going to sound awful, sorry, I don't work with animals because I think it's wrong. But if you have an infected mouse and you treat it with antibiotics, you can literally grind the whole mouse up and prove that the bacteria are gone.
Right.
But if those mice still have symptoms, like you can poke them with a little stick and they twinge and they urinate more frequently
Hmm.
after having a cleared UTI, it does make you wonder, really. I mean, mice aren't people. They're very different in some ways, but
Hmm.
there is some evidence that even a cleared infection can still leave you with lingering inflammatory symptoms, which is disturbing, really. Uh,
Very.
this is why we need to know more about it because you might
Yes.
have two classes of people. You might have the bona fide chronic UTI, recurrent UTI, it's not
Hmm.
going away, it's embedded, it's in a biofilm, you can't detect it. They need to be treated somehow. And
Yeah.
then you might, in an antimicrobial way, and then you might have another subset of patients who have a post infection inflammatory response. And maybe those people need something else like steroids or
Hmm.
another way to sort of dampen the immune system, and it's totally not antimicrobial. And we have no way, because the tests are so bad, you
Yeah.
can't grind up a person and prove they've got no bacteria like you can with a mouse. Sorry to gross people out. I shouldn't talk like that. Really, it's how on earth can we tell the difference between these two types of people without more research into it?
Yeah, and I think that kind of highlights one of the problems that the same approach is used regardless of your experience. We don't have good guidelines for how to treat these patients that have had the problem for such a long time. But maybe you could talk a little bit more about why antibiotic treatment does fail for so many people and recurrence happens, or these chronic infections where people have continuous symptoms.
Yeah, so there's obviously there's a sort of a continuum of recurrent or chronic UTI. You get people that get massive UTIs and then nothing, and they're
Hmm.
totally fine for a couple of months. And then you get people with sort of constant low-grade symptoms. I think these are all in the same spectrum of bacteria that are not responding to antibiotics very well.
Hmm.
And there's so many reasons why. I could just hit a few
Yeah.
scientific reasons why. So you've mentioned a few already because I'm really pleased by how well educated the patient population is. You all know about embedded infection.
Yeah.
So these bacteria are very good at diving into the bladder wall, taking up residence inside the bladder wall where they cannot be cleared by either antibiotics or the immune system. And actually, usually antibiotics work in conjunction with the immune system. You need kind of a healthy immune system and antibiotics to really
Hmm.
clobber it. But if they are intracellular, as we call it, or inside the bladder wall, there's nothing you can do until they eventually will turn over. The bladder's like a conveyor belt, so the cells at the bottom move up and eventually they get shed
Hmm.
into the urine. Eventually, I suppose, if you treated somebody long enough, you might theoretically get rid of all those reservoirs. But most most people only get 3 days of nitrofurantoin if they're
Yeah,
lucky.
it's not a lot.
That's not going to happen. And then you have the case where you mentioned the biofilms. We can
Hmm.
have, and we do see biofilms in human cell models. So we know that they're there. Even without a catheter, we know that you can get biofilms in the bladder. And biofilms are notoriously resistant to antibiotics, not just because they have this slimy outer layer that prevents the drugs from getting in, but when you're biofilm, you're kind of asleep. You're not dividing, you're just sort of hanging out, you're dormant. And almost all antibiotics work by targeting some active process like cell division or metabolism. And if your bacteria is not metabolizing, even if the antibiotic gets to it, there's nothing to act on. So biofilms are a particularly difficult nut to crack. So that could explain recurrent infections. Then you've got, of course, this whole range of really weird and wonderful things that bacteria can do in the presence of antibiotics. If they're just floating there in the urine and they're exposed to antibiotics, some of them can sort of get around the antibiotics for a while by switching their genes on and off. And then when they do that, they can sort of temporarily not get killed and live to fight another day. So the minute the antibiotics are gone, whoop, they're back. And they go back into their normal mode. This is called resilience. It's really hard to study. And it does happen with lots of different bacteria. So we're pretty sure it's happening. But yeah. So those are three major reasons, I think, why people might respond. And of course, there's a fourth one that people don't often think about, which is your own genetics. As I mentioned, you need a healthy immune system as well as drugs to really work together to clear a really embedded infection. And some people may not have the best genetics for that. We know that UTIs can run in families from mother to daughter. It's been shown that there's some mutations in particular genes, especially genes that are sort of sentinels that look out for bacteria. Mutations in those genes can lead to a reduced immune system, which might give you problems. And of course, people who are immunosuppressed, like patients with renal transplants, for example, they get loads of UTIs. So that's another reason. Maybe your immune system is just not up to scratch. And then again, this sort of goes back to your point that we need to know why the UTI is not being cleared to treat it properly. So if it's an immune problem, then you might want to consider immunotherapies or something else that doesn't actually, unfortunately, exist yet. But at least you could do research and try to find a therapy for that problem.
It would be good if there was more investigation into root cause for different patients, because we see so many different experiences in our community. And a lot of people ask whether it's a reinfection. So are they having an infection constantly seeding from their gut, or could it be something in their environment? Do you think there's how many people do you think will be in that boat versus those who might have the chronic infection that is just not being cleared properly?
I think that all of those scenarios are completely possible. You could have people who have a reservoir in their gut and they keep getting reseeded. That's reinfection. You could have people with a reservoir in the vagina, especially in postmenopausal women who don't have estrogen. It's a nice sort of cozy place for them to hang out and regroup, because being in the bladder is no picnic. You know, you're in this place and 6 times a day or 12 times a day if you have a UTI, you're getting flushed out. So the vagina is a nice safe place where they could hang out. They could be hanging out in the bladder wall, another nice safe place. But telling the difference between those, you know, you could do tests on stool, on poo, and sort of compare what's in poo versus what's in bladder. And people have done those studies, and they have shown that people who get UTIs, it can be preceded by this massive bloom of bacteria in the gut. So yeah, that could be the case as well. And I don't even want to suggest it's down to bad hygiene. I mean, I think that particular accusation has been weaponized against women.
We need to put it behind us, definitely.
It's really hard to control the movement of bacteria through a tiny little space, you know, from anus to urethra, where there is, you know, lots going on. You might be having sex. You might be, you're wearing jeans, or sort of rubbing. You're riding a bicycle. You're just walking. You're just existing. And of course, you know, it's not that you haven't wiped from front to back and taken a shower. Like, you know, there's been some really shocking things levied at women and their hygiene regimen, which really makes me angry.
Yes,
it's really hard
from the real problem, which is that we haven't done enough research to know why this is an issue for so many people and how to fix it.
If a man had a urethra that was 1 inch from their anus, they'd be getting lots of UTIs too. And maybe this wouldn't even be a problem because there would have been a lot more research.
They probably wouldn't be blamed for it either.
Yes.
But speaking of treatments, and what do you think the future of UTI treatment will look like?
Well, it's, you know, there's some exciting things on the horizon. I can touch, I mean, your community is so well informed. I think the friendly bacteria angle is really strong. I am a firm believer that the microbiome may have the power to cure all ills, maybe not on its own, obviously not on its own, but if we can maybe engineer it to be a little bit stronger or to synergize with antibiotics. There are some antibiotics that don't kill lactobacillus by nature. So lactobacillus is naturally resistant to, for example, trimethoprim. Not used so much anymore, trimethoprim, but it doesn't kill lactobacillus. So you could imagine some sort of therapeutic scenario where you get a probiotic lactobacillus in conjunction with an antibiotic that doesn't kill lacto, and maybe they can work together with your immune system to be more efficient at clearing bacteria. So
And I
that's one
think
possibility.
patients would be quite interested in that approach too, because we do get a lot of questions about probiotics and how to rebuild the microbiome after antibiotic treatment. With all the side effects that come with that type of antibiotic treatment, there's a lot of interest in other approaches.
Yeah, I don't know how much. There have been there have been about, I don't know, a dozen studies with lactobacillus in people, either through oral pills or through vaginal suppositories or
Hmm.
pessaries. And I think two out of the dozen showed some modest improvement. But none of these studies were there were small studies,
Yeah.
and all the studies used a different bug or a different protocol, so it's really
Yeah.
hard to compare between them. And we're not sure whether the bugs are getting into the bladder. So that
Hmm.
it's been shown, interestingly, if you take a pill, a lacto pill, it ends up in the vagina, which is
Hmm.
not surprising and good news. But does it get into the bladder from there? I would say maybe it would, because we wouldn't have lots of lacto in our bladder as women if it wasn't coming from somewhere.
It has to come
And
from somewhere. Maybe.
it's a vaginal bacteria. So I think but you need to know, what strain do you use? Do you use a
Hmm.
cocktail of strains? You need evidence-based understanding of what is the best lactobacillus to put in there. There's like hundreds of different species, strains
Yeah.
out there. We don't know the best one for this job. And then,
Yeah.
what's the best way to deliver it? Is it really better to get a pessary in your vagina, or do you want to get a catheter and stick it straight in the bladder where it's most needed? Don't know. We need lots of studies like these to work out the best way. But I don't think it's beyond the realm of imagination to think that one day we can cure a lot of diseases, not just UTI, with bacteria that we've engineered to be a bit more effective.
And it
And
likely needs to
what a
be more.
wonderful way, right? It's like,
Yeah.
sorry to interrupt, just because the chances of getting any side effects from this are very slim. And it's like harnessing something natural just to make it a little bit better.
Yeah, I think patients will be interested in that type of approach, but it likely needs to be more personalized because every patient is coming from a different history with a different microbiome. So that could also be an issue with some of these smaller studies. They're not stratifying patients into groups,
No,
they're just looking at everyone as the same patient and applying the same method. But we hear a lot of anecdotal evidence in our community that people have found benefit from using certain types of probiotics. So I think it is worth that research being done.
yeah, absolutely. We just need more research. The bar is pretty low, actually, for if you want to study a new medicine, a novel
Hmm.
drug, it takes the regulatory hurdles are really high, but to study probiotics, they're kind of in a special category, and it's easier to do trials with them, especially if they're categorized as nutritional supplements.
Hmm.
You don't really need much to do those things. But of course, the nutritional supplements might not be the best combination.
Right.
So I
But
don't know.
hopefully we'll see more in that space then.
Yeah, I mean, other things to think about. I think the Euroimmune vaccine looks quite promising.
Hmm.
There's been some really promising clinical data. It doesn't seem to be available on the NHS yet in this country,
Not
but
yet.
in other countries, it's being used widely. It's being used in trials here in the UK and including in my department.
Hmm.
So I think it might be rolled out at some point.
That would be
And
great.
there was some really amazing, so this is not peer reviewed, but it was reported at a conference a few years back, a 9-year follow-up study
Hmm.
of patients on Euroimmune. And just, it was really interesting. Half of the patients responded amazingly well, and the other half, nothing. So
Yeah.
this goes back to your point,
Hmm.
actually, Melissa, that different patients might need different things, but how exciting that half of the patients after
Amazing
9
result for
years,
them. Yeah.
yeah. So,
I
so
think it's
yeah,
worth trying, but it would be good if we could identify what types of patients it would likely help, because at the moment it is hard to access and it's expensive. And so we hear a
yeah.
lot of mixed feedback from our community, but a lot of people who found it quite beneficial.
Oh, really? That's good to hear because I've not really heard. I've heard rumors that, oh, it didn't work very well
Mhm.
for really people who are really bad embedded infections. It didn't do much for me. I heard one or two people say that, but I've never heard anyone saying, actually, it's all right. So you have heard positive reports, have
We've
you?
definitely heard both
Oh, that's
sides.
great.
We have had many positive reports. So even people who say it reduced the frequency of their UTI, which is a big deal for quality
Yeah,
of
yeah.
life.
Yeah, so that's amazing. I'm glad to hear that.
Yeah.
It's a shame that they had to pay so much and that not
Yeah.
everybody can afford that. But I suppose the NHS really needs to, they have to evidence base their choices. But I think there's a lot of evidence now. So I'm not sure what's the holdup, but
Yeah.
I'm not an expert in
We'll keep an eye on it because it is one
NICE and
of
how
the
they approve things.
most frequent questions we get is, can I access Uromune in my country and how? And in most countries, we just don't have the information yet.
I think I Googled it once, and in the UK,
Yes,
you can get a private appointment. I don't know how much it costs. I
quite a
was afraid
lot.
to look. Yeah.
It's quite a lot still. So it would be great if the NHS can take it on board at some point.
If you just make the economic case,
Mhm.
the sheer number of antibiotics that are used, all the sort of clinic visits and tests that are done that are useless, if
Exactly.
you could prevent even a fraction of those, there, I would imagine there's a good business case.
I
But,
think there is.
but I don't know. You'd have to crunch the numbers. But to me, it seems like a no-brainer. I guess maybe the definitive phase 3 trial hasn't been published. I don't know. I don't know what the holdup is. It's not holding
I'm
up
not sure
Europe.
either. Yeah.
They're just all using it now.
Yeah, it's much more accessible in Europe. But speaking of novel therapeutics, what are you working on in that space?
Oh, that's an exciting question. So we decided to target the intercellular bacteria in the biofilms
Hmm.
because we think that maybe not for every patient, but for quite a few patients, that's probably one of the issues. Antibiotics don't penetrate the bladder wall. Some do. There's a few classes
Hmm.
of antibiotics that can, but they're not the ones that are normally indicated for UTI. So we thought, wouldn't it be great if you could take a bog standard, really decent antibiotic like nitrofurantoin, which
Hmm.
has an excellent safety profile and has been used for almost 60 years, and the resistance is really low because it has multiple modes of action. Whenever a drug has more than one way to do something, the bacteria struggle to resist it. So nitrofurantoin has an excellent profile. And the only safety issues it has to do with issues with the lungs. So, but that's
Hmm.
because you're taking it as a pill and your entire body is being dosed. And of course, your entire microbiome is being dosed by
Yeah.
nitrofurantoin. So you might get stomach issues, you might get lung issues that are quite serious actually. But what if you could put the nitrofurantoin directly into the bladder and make it penetrative enough so it could go into the bladder wall and kill those bacteria and bust up the
Hmm.
biofilms while they're at it? That's what we thought. We thought that'd be a great idea. UCL was working together with Oxford on this, and we quite some time ago, as you probably know, we came up with this solution called capfuran,
Hmm.
which are microencapsulated nitrofurantoin little spheres. They're about 5 microns. For reference, a bacteria is about 2 microns. So
Right.
if you were a bacteria, capfuran would be the size of a car. Right? So it's a
Okay.
massive thing. So these are not intended to go into bacteria. They're
Right.
intended to go into cells, and the cells then, so they're like a balloon full of antibiotics, and
Hmm.
they float down, dock onto the bladder wall, and then they push the drug into your cells and through the bladder urothelium
Hmm.
without the capsule going in, which is great because the capsule is made up of, although it's a biodegradable, non-toxic polymer, you really don't want that stuff gumming up your cells. So
Right.
it's great that they just. And if you watch a video of this, it's really cool. These capsules come in and they just if you put some green dye in there to follow what's happening, they go and they squirt it in, so the cells all turn green, the capsules turn clear because they've emptied, and then of course they will be urinated out in your next wee, or they'll
Hmm.
biodegrade. And the nice thing about this, I should say, and I should have said at the beginning, I have stock options in the spin out company at UCL.
Yeah.
So I take what I say with a grain of salt because I have a conflict of interest. But personally, having said that, I think it's a really nice idea. Have really promising preclinical studies. And yeah, the idea is that the bacteria cannot escape from the drug if it's being delivered in this way.
Is that something you would take orally or via a
No,
catheter?
unfortunately, it's what we call an intervesical
Hmm.
treatment. It's a catheter, which is right, not great. But on the other hand, you will not be dosing your lungs or your gut microbiome with this nasty antibiotic.
Yeah.
So there's a trade-off there. yes, it's a little bit uncomfortable, but we hope that you don't need more than a couple of squirts of this. We don't know the regimen yet,
I think a lot of
but
people are okay with that kind of treatment. We get many questions about interval cycle treatment options, and there just aren't a lot for this patient population, and they would be interested if it
yes,
was successful.
they already get gag, hyaluron, and
Hmm.
these are the gag installations, and some of them get intravacycle gentamicin treatment.
Yeah.
So we actually did some market research because we were curious. We got a grant and we did a big survey in the US and the UK, just simply asking people, if you had a treatment that worked, would you mind having a catheter?
Hmm.
And most people are like, bring it on,
Yeah.
if it
Yeah.
actually works. Yeah, the acceptability seemed to be quite high. So yeah,
That's
that is
probably
the
true.
thing. We are doing really well, but the problem, of course, is funding. It's
Hmm.
very hard to convince investors that this women's disease that's not
Yeah.
breast cancer, it's not killing lots of people. It's hard to convince people that they'll make a return on their investment.
Hmm.
So sometimes I go to these investor meetings and it's a whole sea of Hugo Boss suits, and there's me up there trying to convince them that this is important. I usually manage it because I say, think of your girlfriend, think of your wife, think of your grandmother. You know,
Hmm.
there's a lot of people suffering from this. And I think, yeah, we have some really wonderful benefactors. We
Hmm.
just haven't cracked that last funding round that we need to do the clinical trial. Also, we are manufacturing this product in a factory,
Okay.
which has proved to be difficult initially to scale up this to. And we were doing it in a lab on a benchtop, and now we have to make a huge
Yeah, it's a
batch.
big
And
change.
yeah, it's taking a while, but I think we've cracked it now. So
Okay.
we're just now putting together, hopefully, a nice package where we can raise some funds
Have you
and get this
tested this
going.
in humans already, or is
No,
that what your next step will be?
no. So, but the good thing about this, from a regulatory point of view, is it's only two ingredients:
Hmm.
nitrofurantoin, which of course is a generic,
Yeah.
and the polymer, which is already in use in people as a contrast agent. So if you ever had an MRI scan, you may already have had this polymer. So it's already approved for FDA approved, MHRA approved. So the
Great.
two ingredients separately are approved.
Hmm.
If you think about anything that goes into a bladder, compared with putting it in your bloodstream or your gastrointestinal tract, it's a privileged environment. What happens in the bladder stays in the bladder.
Hmm.
Things don't get out of there. So even if they were mildly toxic, which they aren't, they're never going to get out of the bladder space and into the bloodstream. So I think it's got a really good chance of getting a swift, if it works, getting a sort of a swift regulatory approval. We
Yeah,
really
and I
hope.
think the patient reception for this type of therapeutic will be good as well. So if there's any investors watching this video, maybe they can reach out to you. What is the best
Yeah,
way for someone to get in touch with you?
just email me. Just Google me. I'm all over email. Just look at UCL's website. You can get me on LinkedIn. You can get me on socials. Yeah,
And
that'd be
have you
great.
published any papers on this capsule?
Yeah, it's been published a few years back. You
Hmm.
can read about it.
So if you can share the link with me to
Yeah, if
any
you have
papers, I'll put
sure,
them in
sure,
the video description so people
sure.
can find them easily.
Yeah, so we tested this on our human bladder mini bladder, or sort of our mini bladder model, which is quite
Hmm.
exciting. And we didn't need to use any animals to test this because the two ingredients are so well known. Unfortunately, there does need to be a toxicity study in rats.
Okay.
It's mandatory. We can't get around it, which makes me sad because I don't tend to work in animals.
Yeah.
Uh, but yeah, just to prove that obviously you don't want to put something in a person that hasn't at least touched a few rats, I guess.
Yes. So we hear that about most things going to trial. So I suppose it's
Yeah.
unavoidable.
But it's actually good that we don't have to test the efficacy in an animal model because I have to say the animal models for UTI are terrible. I probably have some mouse people listening, maybe not terrible, but there's a lot of difference between humans and mice in the
Hmm.
bladder. And imagine you had a brilliant new drug and you were forced to test it on animals to prove it worked, and it didn't work. But actually, it would have worked in people. This has happened
Hmm.
quite a few times, apparently, and vice versa. You've had something that was really highly toxic in mice and never went any further, but
Right.
in a human, it wouldn't have been. So the mouse, as a gatekeeper, has problems. So I was very pleased that we were given basically. It seems that we'll be able to just circumvent that kind of test and just use our human mini bladder as the ultimate arbiter of whether it's safe and effective. And then the inevitable rodent trial just for the toxicity, which we can't get out of.
If you can get the funding in place, what's the timeline, do you think, until clinical trial?
Yeah, so not too bad. I think we could even be looking at next year if everything lines up.
Okay,
Of course,
it's
you need
very
to get
soon.
you need to get approval and the funding and the approval, and 2026, maybe 2027, I hope.
Okay, well, it's
Although
exciting.
we have thought that before, and think, things, COVID really set us back.
Hmm.
There were very few, interestingly, very few manufacturers of nitrofurantoin. Most of them
Right.
are in India. The supply chain was, especially during COVID, was just a nightmare. And yeah, it's interesting how interdependent all the countries are when it comes to drugs, because people don't really
Hmm.
think about it, but many of the things that we take from our GP are produced in other countries, and supply chains make a big difference.
Yeah.
For example, a few years ago, we were having massive problems getting HRT. I think it was estrogen. There was this massive
Hmm.
shortage. Women were meeting in petrol stations to
Yeah. It's
exchange
been
spare
terrible.
pills. It was awful. That would never happen with Viagra, I have to say. But
That's
yeah, you
for
can have
sure.
supply chain issues, and the pandemic was really terrible for our trajectory. It set us back a few years.
Hmm.
But I think we're back now,
Well, that's
and
great to hear.
hopefully, if we can get the funding, hopefully it will fly. And of course, it may not work, right? Things may not work as you intended.
Hopefully
But
that's not the
I have a
case.
good feeling, but I can't say for sure.
Yeah, well, it's great to hear the updates on that because we often get questions about what you're working on. The patient community is quite aware of what you're working on and very enthusiastic about joining a clinical trial once you get there. So I hope that you can get the funding in place and maybe this video can help. And we're also happy to share any papers that you publish or anything else that might help to raise awareness about what you're working on.
That's really kind.
Really wanted to thank you again for chatting to me today and answering all the questions that we received.
You're very welcome.
Thanks so much for watching. I hope you found this expert video helpful. If you'd like to learn more about this or related topics, be sure to check out our other videos or head over to liveUTIfree.com for related articles. We'll drop some links in the video description. If you like what we're doing on this channel, you can support our work by hitting subscribe here on YouTube, and don't forget to tick the bell so you'll be notified of our future videos. Thanks again for watching, and until next time, keep asking questions and pushing for better solutions.